Dreams, they take us places we've never been, show us things we've never seen. Dreams let us reimagine the world we live in. There's a power inside us that can make dreams come true. The power of the genome. We found a way to unlock that power so you can answer life's biggest questions. Can you find a cure for every kind of cancer? Can disease stop being an inheritance? How can everyone live not just longer, but healthier? These answers are written just 4 letters. We live in a time that is brighter than ever before, and this is just the beginning. This is the genome era.
Illumina first brought NGS to the benchtop more than a decade ago and continues to set the standard for simplicity and speed. The MiSeq i100 series. The series offers 2 systems. The MiSeq i100 system enables 15 gigabases of data output with 25 million reads. And the MiSeq i100 Plus system has an expanded output of 30 gigabases with 100 million reads. There are 10 reagent configurations for flexible outputs, providing deeper sequencing or the ability to run additional samples for a variety of applications, including small whole genome sequencing, pathogen detection, and targeted gene sequencing. The MiSeq i100 series is simple at every step, from setup to data analysis. The reformulated reagents are shipped and stored at room temperature, eliminating the need for freezers. There's no need to wait to thaw your consumables before starting a run. Run setup is performed in 3 steps. Dilute libraries, add denaturation buffer, load the cartridge. The instrument will walk you through loading the cartridges, minimizing the chance of user error. After a few minutes, the pre-run checks will complete and automatically initiate the run. You can monitor the progress of the run on the instrument or the cloud via a laptop. XLEAP SBS Chemistry, our fastest, highest quality, and most robust sequencing by synthesis chemistry, enables a 4-fold reduction in turnaround time compared to the MiSeq system, providing same-day results. Onboard Dragon offers accurate, comprehensive, and efficient data analysis. Data analysis has been simplified with sample-to-analysis workflow solutions, generating results in 2 hours. Through Base-Space Sequence Hub, you will have access to the full suite of Dragon analysis pipelines for accurate, rapid data interpretation and visualization, minimizing the need for NGS bioinformatic expertise. Following the completion of the run, remove the cartridges from the instrument. The cartridges can be easily disassembled, and a majority of the components can be recycled. This is the highest standard in benchtop sequencing. This is sequencing for every lab. This is the MiSeq i100 series.
Muy buenas tardes con todos y bienvenidos a nuestro evento del día de hoy, Future Medicine, Transforming Global Health with Precision Gene Editing. Muchísimas gracias a todos los que se han unido a través de nuestros 2 canales desde YouTube y a través de Facebook. Agradecemos su presencia porque creo que este evento va a marcar pues, el inicio de algunos que son parte del Instituto de Investigaciones de Biomedicina. Este evento precisamente reúne a destacados líderes en el campo de la edición génica para explorar cómo estas tecnologías están transformando la medicina y abriendo un mundo de posibilidades en la lucha contra enfermedades complejas, como lo que es el cáncer, la diabetes y otras patologías genéticas. Es así que la edición génica es la clave para un futuro donde la medicina no solamente trate, sino que cure con precisión y eficiencia. Y todo lo que estamos haciendo, todos nuestros esfuerzos por parte del Instituto de Investigaciones en Biomedicina, buscan precisamente generar la evidencia necesaria para poder pasar de estas aplicaciones que se generan dentro del laboratorio hacia lo que es el paciente. Este campo es interdisciplinario y es un gran gusto pues de estar con ustedes el día de hoy y presentarles estos avances por parte de la universidad y justo con un invitado internacional muy importante. Empresas como GenLife, con su compromiso en proveer herramientas de la última tecnología, y organizaciones como el iBiomed promueven esta innovación científica. Quisiera pues comenzar agradeciéndoles a todas las personas que están conectadas y a su vez a nuestro patrocinador, quien es GenLife. Voy a dirigirme justamente en unos instantes hacia ustedes presentándoles un poco de lo que hace GenLife, que es una empresa especializada en la comercialización de equipos reactivos y consumibles de última tecnología para biología molecular. Ellos son representantes de Lumina, una de las marcas líderes, como pudimos ver en uno de los videos al principio de nuestra transmisión a nivel mundial en lo que es la secuenciación. GenLife promueve el desarrollo de la investigación genómica en nuestro país, facilitando el acceso a los laboratorios locales y herramientas innovadoras para la secuenciación de alta precisión. Esto abre sin duda las puertas a nuevas oportunidades para los estudios en biomedicina y muchos otros. Quisiera pues ya invitar a nuestros speakers, a nuestros presentadores, a lo que sería el stage, y agradecer también a Andrés y a Pierre, que están justamente tras cámaras y que son pues nuestro punto de apoyo a través de marketing digital y su transmisión. ¿Cómo estamos? Hi, Victor. How are you? How's everything? Victor, I'm sorry, you're muted.
That's great. Thank you for inviting me.
No, we
It's
are
an honor
super
to
happy.
participate in this forum.
Excellent.
Hello,
Hi,
everyone.
Carlos. How's everything?
Good. I'm doing good. How about you?
Well, now that we are starting this webinar that we were planning it and expecting it for so long, this is such a nice experience. And from the Institute of Biomedicine, which was founded a couple of years ago, is a great opportunity for us to start these webinars. I hope that we are going to have a lot of them, and also to show some of the latest results that you have shown so far in incredible research that you are doing in Rogers and also at the USFQ. Great. So let's move forward. Voy a presentar primero a Juan Carlos Collantes. Justamente él es nuestro jefe de innovación en edición génica en la universidad y es un experto en biotecnología y edición genética aplicada en la salud. Es profe del Colegio de Ciencias Biológicas y Ambientales. Tiene un PhD en biociencias moleculares de la Universidad de Rogers y ha desarrollado tecnologías tan increíbles como el Pinpoint, y que esta justamente fue galardonada con el Edison Patent Award. ¿Podrías darnos algunos insights de lo que fue el hecho de ganar este tan importante reconocimiento, Juanca?
Sí, bueno, muchas gracias, Andrés. Realmente esta fue una grata sorpresa que tuvimos hace un par de años. La Universidad de Rutgers nos nominó a la oficina de transferencia de tecnologías en Rutgers. Nos nominó al Dr. Jin y a mí para participar en estos premios que son otorgados por el Consejo de Investigación y Desarrollo de New Jersey, que es avalado por el Congreso del Estado de New Jersey. Y competimos con gente, con empresas como, por ejemplo, Siemens, Merck. Habían otras universidades como Princeton, el Instituto de Tecnología de New Jersey, estaba Johnson & Johnson después de desarrollar las vacunas. Entonces estuvimos compitiendo con líderes en el área de biotecnología y nosotros resultamos galardonados en el área de biotecnología por el desarrollo de una tecnología de edición de bases basada en aptámeros de ARN que les vamos a contar un poco hoy. Entre mi presentación y la presentación de Víctor vamos a contarles un poco sobre cómo funciona la tecnología y qué es lo que hemos hecho para aplicaciones al respecto en torno a la salud humana.
Excelente. De hecho, no podemos esperar, y qué gusto también tener a Víctor. Entonces, dear Victor, I will introduce a little bit about your amazing background. So, Víctor es profesor de la Universidad de Rogers y es miembro del Instituto de Cáncer de New Jersey. Es el líder en edición génica de precisión y tiene, pues, junto con Juan Carlos, ellos han desarrollado esta plataforma que es el Pinpoint y que está, pues, fue desarrollado para lo que es de combatir el cáncer y enfermedades genéticas. Es un gran honor el hecho de poder contar con ustedes el día de hoy en este webinar que es gratuito, entonces para que todo el mundo lo pueda pues disfrutar y que sea un una base para el desarrollo de nuevas tecnologías. Entonces, ahora sin esperar mucho más, pues vamos con Juan Carlos Collantes y tu excelente presentación. Muchísimas gracias.
Gracias. Oh, ¿pueden ver mi pantalla, Andrés? No sé qué está pasando. Ok, voy a dar esta presentación, la voy a dar en inglés, justamente porque estamos con el Dr. Jin. Entonces, el formato va a ser que voy a hablar sobre la tecnología, voy a hablar al respecto de nuestra tecnología, voy a hacerlo en inglés, y luego, al final de la charla de Víctor, podrán hacer preguntas que pueden ir escribiendo en la plataforma. Okay, so first of all, I would like to start by thanking everyone for being here, for sharing this time with us, and I would like to especially thank the organizers, the Institute of Biomedical Research at USFQ, for inviting us and for allowing us to share our developments in genome editing technology. And for me, it's a great pleasure to share the stage with Dr. Victor Jin, who is one of the greatest scientists that I've had the privilege of meeting. I've been working with Dr. Jin for the past decade. I joined his lab as a grad student, later as a postdoc, and together we developed a technology, a next generation genome editing technology that's called base editing, with which we can transform the genome without breaking the DNA. So first of all, CRISPR, which is the basis of this technology, the foundational technology of base editing, CRISPR is a revolutionary technology that has been applied to edit genomes all across the tree of life. Even the people are trying to bring back extinct animals like the woolly mammoth. So this technology is very powerful. It is a pair of molecular scissors that can be programmed, guided by RNA, which is this red strand, and you can program it to target virtually any genome you would like. And once it engages with the target, when it finds its target, it activates the molecular scissors, inducing a double-stranded break in the DNA. These, in turn, activate the cellular response to DNA damage, and the repair will introduce genome modifications. So this technology was developed over 12 years ago, but these ladies, on the right, we have Dr. Jennifer Doudna, and on the left, we have Dr. Emmanuel Charpentier, who developed this technology to be used as a genome editing tool. Their work was so groundbreaking that they were awarded the Nobel Prize in Chemistry in 2020, just 8 years after they first described this technology. So this, of course, led to a lot of excitement in the field, because for the first time, we could dream of really being able to cure the incurable. Basically, this is a paradigm-shifting technology that will allow us to go to the root cause of genetic diseases and other complex diseases like cancer and correct it at the root, right? So a lot of papers came out after the discovery of CRISPR deploying this technology to correct genetic diseases from sickle cell anemia to CFTR, even cancer. Right. So actually, last year, the approach to treat sickle cell anemia and beta thalassemia was approved by the FDA. So it's the first time we have this type of technologies being approved for human use. However, you know, the more and more people were working with this technology, people started to realize that it could also aside of being a very powerful and efficient tool, it could also lead to unwanted outcomes like genome instability that would lead to all sorts of chromosomal mutations like translocations, deletions, inversions that would lead up to cancer. So it has oncogenic liability. The thing is that if you are trying to treat and cure a patient from a genetic disease and then you give them a cancer, it's not ideal. So there's where we join this race. So when I started my grad school at Rutgers, my graduate studies, I joined Dr. Victor Jin's lab, and he already realized that this particular problem, this double-stranded break-induced genome instability, could be a hinder to advance this technology to the clinic. So when I joined his lab, the challenge was to create a system that could change the sequence of the DNA without breaking it. And it was not, any trivial challenge, because for the past 20 years, previous genome editing technologies, prior generations of genome editing technologies, all were based on the induction of double-stranded breaks. So we had to think of ways in which we can modify the genome without breaking the DNA. So it turns out that three groups were independently working on the same problem. On the one hand, in Japan, there was a group at Kobe University, and Massachusetts, at Harvard. There were another group that were also developing this, and then us at Rutgers, we all three were independently developing a system that was later dubbed base editing, that could lead to sequence modifications without changing the DNA, without breaking the DNA, I'm sorry. So basically, you can program this, say this is the target site, and you can program these gene editing tools to target a region in which we can induce a point mutation based on a deamination event. So these technologies use cytidine deaminase enzymes that can eliminate this little chemical group, which is an amine group in the cytosine residues, and turn them into uridine, basically changing the C to T in the DNA. These can be used in many ways to edit genomes, transform genes, knockout genes, or correct pathogenic mutations without actually breaking the DNA, because the Cas9 molecule here has been genetically engineered to avoid generating double-stranded breaks. So basically, what we do is we recruit this effector molecule to the site by CRISPR to induce this point mutation, right? So our system is a modular RNA-mediated base editing system that has 3 modules, right? Here we have the genetically engineered Cas9, which is part of the CRISPR system. This has been engineered to avoid breaking the DNA. On the other hand, we have a second module, which is the gRNA module. So it has a chimeric RNA molecule that can be programmed. This little yellow part here can be programmed to target any part of the genome, any site of the genome, right? So basically, here's like you can program the genomic coordinates that you want to target. This blue portion is the CRISPR motif. Basically, it recruits this enzyme, right? It interacts with this enzyme. And this red part is, these are RNA aptamers, which are non-coding RNA molecules that can be used as molecular magnets to recruit an RNA binding protein, and a specific RNA binding protein, which in turn is fused to a cytidine deaminase effector. Basically, when these assemble on the target site, these little red aptamers can recruit this effector module and change the cytosine to thymidine by removing this chemical group. Basically, base editing induces a precision chemical change on the DNA, changing the, creating a point mutation without breaking the DNA. So I mentioned that other groups were developing the same technology, and the two surviving technologies are one at Harvard and the other one, our technology at Rutgers. The difference in architecture between these two technologies is basically that the Harvard system, it directly fuses the Cas9 molecule to the cytidine deaminase effector by a protein linker. So it basically this deaminase is tethered to the Cas9 protein, right? In our case, the effector module and the CRISPR module are separated, and they are recruited on the site of action by these aptamer RNA binding protein interaction. So this is important. This modularity is important, as I will show you later. So first thing we thought is, how can we make this work? So we turned to the molecular biologist's favorite gene, GFP. This is a fluorescent protein that we borrowed it from a jellyfish. And this gene, when this protein has a fluorophore, basically is a sequence of 3 amino acids, a threonine, a tyrosine, and a glycine, that when the protein folds, they emit light. So if we make a mutation here in this particular base pair, this AT base pair, and change it to GC, we can change this tyrosine to cysteine, basically changing the sequence of the fluorophore and therefore eliminating the fluorescence. So basically, by doing this, we create a proxy of a human or a genetic disease of a loss of function mutation. So the protein is getting synthesized, but the fluorescence is not coming out. So basically, the function is lost. So the idea to create this mutant gene was to then restore the fluorescence by base editing. So basically, the idea was to generate this mutant protein. We confirmed that the cells were synthesizing the protein, but the protein was not fluorescent because of this point mutation. And upon treatment with our RNA aptamer base editing system, we restored it to a fluorescent state, basically curing this loss of function mutation, restoring the function of the protein. This led to a licensing agreement between Rutgers and Horizon Discovery, part of the Perkin-Elmer group, and they are now marketing this technology as that now is known as pinpoint base editing system. So they are developing therapeutic applications. So we thought, so now we edited a jellyfish gene, basically generated a proof of concept that the system can work on eukaryotic genomes, but we needed to try to test this technology, in human cells with therapeutic value. So the first thing we did was to, leverage the system orthogonality. So when we say orthogonal, we mean in a biological system, an orthogonal system is a system that one part of the system will not affect the activity of another. So basically what I'm trying to say here is that because the recruitment of the effector module is mediated by this particular molecule interacting with this one, we can just change this to a different pair and recruit a different effector protein. So we can use, for example, an AID. AID is a cytidine deaminase enzyme from the human adaptive immune system. We can change it for another type of cytidine deaminase called ApoBec, or even we can use adenosine deaminases that can change from A to G in the genome. And even we can avoid base editing by just not using the aptamers, and we can induce a controlled double-stranded break for an insertion of a transgenic gene. So we thought to ourselves, how can we leverage this modular and orthogonal design for human health? So for this, we turned to immunotherapy, specifically CAR T cell therapy, in which people people with certain types of cancer can be treated with T cells that are extracted either from the patient or from a donor. They are reprogrammed genetically to train these cells in order to recognize cancer cells by introducing a chimeric antigen receptor. That's where this name comes from. Chimeric antigen receptor is an antibody that is raised against the a tumor antigen. So we multiply these cells in the laboratory, and then we can treat the patient with these newly edited cells that now are programmed to destroy cancer cells. So the thing is that we had this idea, and a very interesting collaboration ensued after this between Rutgers, Horizon, later Revity, AstraZeneca, and us at USFQ from the biotechnology department. We, recently published a paper in which we described a method to generate CAR T cells by simultaneously knocking out several genes and at the same time introducing this CAR cassette that expresses this chimeric antigen receptor, this antibody that can lead the T cells to the tumor site. So this has to be done in one single step. So the 4 loci that were selected for this study are these 4 genes: CD52, PDCD1, TRAC, and B2M. These are all genes that have been shown to generate robust CAR T cells, and I'm showing the genomic sites that we targeted here. So we had to induce 4 gene knockouts, meaning that we had to eliminate the expression of these genes. So how can we do that? We can either generate point mutations on codons that encode for glutamine, arginine, or tryptophan, changing these cytosine and the complementary cytosines here, and turn them into stop codons, premature stop codons that will delete or destroy the expression of this gene, therefore making this gene silent, right? Alternatively, you can also modulate the splicing mechanism by mutating these, the 5' splice site or the 3' splice site, therefore inducing exon skipping or exon intron retention that would ultimately lead to frame shift mutations that will end up truncating the protein, therefore eliminating its expression. So in the study, we used these 2 approaches to generate these CAR T cells. So all the experiments were done at the Horizon laboratories in Cambridge, UK. So basically, the experiments performed were multiplex editing of these 4 genes, which we are seeing in these black graphs, that show very efficient C to T conversion in the target sites that will induce gene knockout. And we can see here in this other graph, we are showing here the percentage of protein loss. Basically, protein loss is it's the expected outcome after gene knockout. And we can see that the levels of gene knockout by protein loss are comparable to the conventional Cas9 system, right? So aside from the gene knockouts, as I said, right, we did the knockouts by base editing, we also need to introduce the CAR cassette. So we did that by generating a controlled double-stranded break with two guide RNAs, targeted to a specific loci, a specific locus, without recurring deciding deaminase effectors, basically generating a single strand break here, another strand break over there. These offset nicking will induce a double-stranded break, and we compare this to the conventional double-stranded break genome editing by wild-type Cas9. So we compare these two approaches, and we see that, again, both our RNA aptamer, our base editing system, right, modified here, can induce comparable levels of CAR cassette insertion. This is a knock-in, right? So we are knocking in this CAR cassette with a comparable efficiency to Cas9, to wild-type Cas9. So the thing is that, you know, because I mentioned earlier that these interventions in the genome, basically the ones that induce DNA breaks in the genome, can lead to all sorts of unwanted chromosomal outcomes. We tested, you know, whether these interventions in these T cells would lead to translocations. So the scientists at Horizon did very interesting experiments in which they surveyed the genome, and they found that there were many translocations among the 4 loci that were targeted, right, by the conventional CRISPR system. So this circus graph right here is showing the balanced translocations that resulted after a wild-type Cas9 experiment. So that's what we are seeing here. This is the frequency of balanced translocations shown by after the treatment with wild-type Cas9. But if we did the same experiment, when we do the same experiment for our system and unedited cells, we see that neither of them induce any translocation, basically showing that our RNA aptamer mediated base editing is very safe in order to create CAR T cells. They don't induce any unwanted translocations or any other chromosomal aberrations. So now we are looking into the horizon to see what's next. What can we do next? So here at USFQ, we have several projects. I just show you 3 of them. One of my projects here is aimed to improve on-target precision, basically making these base editing systems laser focused. And we are also applying this technology in order to, tackle a very important problem in public health, which is antimicrobial resistance in pathogenic bacteria. So one of the mechanisms by which bacteria become resistant to antibiotics is by generating an extracellular matrix called biofilm. So biofilm is a physical barrier that does not allow the penetration of antibiotics into this structure. So basically, the bacteria that dwells inside these biofilms cannot be exposed to antibiotics. So the approach that we are developing here at USFQ is that we are aiming several genes involved in the process of biofilm formation in order to see whether these can destabilize biofilm formation and resensitize these pathogenic strains to non-antibiotics. So basically, we're joining the fight against antimicrobial resistance. And on the other hand, we are also expanding the base editing platforms by searching for novel Cas9 orthologs, different RNA aptamers, and different side-editing deaminase or adenosine deaminase effectors that can be used in these systems. And with that, I would like to acknowledge my incredible team of students that work with me. I have 2 teams of very highly motivated students, both undergrad and grad students, and I have these 2 groups. One is involved in improving the on-target precision of base editing. I have David Vaca, who is recently joined my group. He's a master's student. I have Anais Torres, who is a biotech student, Andres Leon, who is graduating next week, actually, from the biotech program. I have also Benjamin Arias, who is also a master's student, who is also graduating very soon from the master's in microbiology. And also we have Vanessa Espinosa, PhD student, and Montserrat Tapia, another biotech student, all of them are working with me to develop these novel biotechnologies. And I also would like to thank my USFQ collaborators, Dr. Melanie Ruiz, Dr. Vanessa Romero, who are collaborating with me in the project for improving the precision of base editing, and also Dr. Antonio Machado and Dr. Sonia Zapata, who are working with me in the biofilm interference. And with that, I will also like to thank all the institutions that have, somehow been part of these projects. A special thanks to Dr. Victor Jin's lab, who I'm constantly collaborating with, and also USFQ. A special thanks to the Dean of Research, the Office of Innovation, the Biotechnology Department, the Institute of Biomedical Research, and the Institute of Microbiology, and the companies that have also helped develop this technology. So with that, I'm done with my talk, and I will take questions if there are any questions at the end of the talk.
Excellent talk, dear Juan Carlos. Amazing technologies that are under development, and it's great to see that the pinpoint technology is achieving great things and moving forward thanks to the interaction between academia and industry.
That's true. That's essential for biotechnology development.
Yeah.
Actually, yes.
So just a little more question. How do you see that? It's like, are we in Ecuador moving forward to that direction? Are we doing the good step? Are we making the right moves?
So I'm, doing my best to get this type of technology, not only base editing. I'm also working on other aspects of biotechnology, like synthetic biology and other fields. I'm trying to push this, but of course it's a bit challenging, here in Ecuador, because of all the difficulties that we have. But it's doable, and luckily I have the support of USFQ. So we are together pushing this through, trying to get these technologies, to fly here in Ecuador.
Excellent.
So I'm optimistic.
Of course, we are all crossing fingers for that.
Yes.
Excellent. So now we're moving forward to our keynote speaker, Dr. Victor Jin, and we are very happy to have you here, and we are amazed by what you've done with Juan Carlos Collantes, and also very proud of. Thank you very much for the amazing presentation and the exciting applications that are coming. Together with Juan Carlos, we had amazing. A lot of questions from the public, and thank you very much to all the people that join us in the live broadcast. And I don't know if Juanca could be also on stage. Juanca, he's around, isn't it? I don't know. We lost Juan Carlos. All right, perfect.
I am here.
All right. I was like, where Juan Carlos are you? Like, yeah. It was not the time to leave. Okay.
No, I'll stay to the end.
Excellent, till the end. Perfect. So we have a lot of questions from the public. So I will pick some a little bit random. So we have one first question for Juan Carlos, and so is from Ana Victoria. Have you found any side effects of the pinpoint technology, maybe changes and unwanted locations?
So every technology derived from CRISPR has this inherent promiscuity. So it can, go to similar regions around the genome that are called off-target sites. and they can induce mutations. But when we have done some of these off-target analysis, we've seen very, very low, even undetectable levels of off-target effects compared to, for example, conventional CRISPR. So conventional CRISPR can go and mutate these sites by breaking the DNA in these off-target sites. But with our technology, we mitigate that almost to undetectable levels. So it has, as any therapeutic agent, it has some secondary effects, but or side effects, but as off targets. But compared to the existing technologies, they are very, very, very low. So it is very safe. And as I showed you guys, the technology, even when you target it directly to a given locus, it does not induce any chromosomal aberrations, which makes it very safe and a very good candidate for clinical development.
Excellent. Thank you. Thank you for your answer, Juanca. And now I have a question for Victor from Nicolas Esteves. In your opinion, which of all the 4 methods of gene editing you mentioned is the most accurate and effective, or they are all equally useful?
So these technology are for different applications. So the conventional is a double-strand DNA break. It's good for inactivation, but it has liability. And the second one, base editing, is for change one single base, and the primary thing is for insertion deletion. And in a way, it is that different. So it's really, you need to understand for apply them, you need to understand what's your goal, and you then choose the right tools to do that. Yeah. So you have efficiency. There's not one that is the most effective and least troublesome, and you have to really use the right tool to address the right need. Yeah.
All right. Excellent. Excellent. Thank you very much, Victor. So now we have a question from Adrianela Cabeza. La voy a hacer en español. ¿Qué ventaja genera el sistema con el fluorophore en su prueba de concepto? Es bastante ilustrativo, efectivo, y si hay otros genes que se podrían probar.
Para una prueba de concepto en un desarrollo tecnológico, normalmente buscas una prueba rápida, que te demuestre rápidamente, y en este caso de forma visual, que está funcionando el sistema, y lo puedes medir, puedes correrle por citometría de flujo, por ejemplo, y medir la eficiencia de la corrección medido en función del número de células que florecen. Pero eso solamente es la prueba de concepto, ¿no es cierto? Entonces hay varios genes que se pueden usar como prueba de concepto. Genes que emiten fluorescencia, como el que usamos nosotros, genes que emiten luz, genes que emiten alguna otra señal que pueda ser detectable. Entonces, simplemente es una parte del desarrollo tecnológico que nos permite a nosotros, como investigadores, entender cómo funciona el sistema, qué tan potente es, qué cambios podemos hacer, pero de ahí obviamente tenemos que mudar el sistema a aplicaciones ya reales. Entonces lo probamos en genes endógenos y luego, como viste, le probamos en genes que tienen valor terapéutico, como por ejemplo para crear células CAR-T.
Perfecto. No, muchísimas gracias, Juanca. Now from Anais Torres to Victor. Are there any structurally or functionally similar systems to CRISPR-Cas9 in eukaryotes? If so, can these endogenous eukaryotic systems be adapted for aptamer-based gene editing?
That's a great question. So one, and I remember many years ago, we were talking about find the human CRISPR. So the long story short, in the mammalian system, we don't have this CRISPR system, but we have a different engine, mammalian engineering system that's right now is heavily intensively explored is called transposon-derived machinery. So in mammalian system, we have different types of transposon-related system. Our lab and other labs are involved in find the value in genome editing, especially by incorporate a large piece of DNA into the genome. Yeah.
Excellent. Thank you. Thank you. Very interesting question, actually. Yeah. So, and we have space for one last questions. It's been an amazing webinar. So from Christian Benjamin Arias, he thanks to all of you, and this question goes to Victor. Perhaps an approximation of how they did the engineering. It is known that the spider cell sequence is repetitive and can complicated and be complicated to use. So this is a question from Benjamin. So so the thing is, it goes through the spider cell sequence. And how easy it is to modify.
I think no, I think the outcome is, and they use maybe in the end you just need to change one. I forgot the detail on how they did the engineering, but they did it, and they replaced a silkworm protein with the spider protein. I think that's in general that's what they did. To me, probably the repetitive sequence, it really doesn't matter if you just clone the cDNA and dump it into the silkworm, replace the silkworm place downstream the promoter, probably will do it. Yeah. So what I'm trying to emphasize is there's a lot of things to be done. And using this technology, it's essentially the creativity and the imagination and of course the need can lead to a lot of very interesting discovery, invention, and applications.
Excellent. Thank you. Thank you very much, Jean, for Victor, for this answer. So we just arrived to the end of our webinar. And I think it has been such an amazing experience for all the people that has joined the broadcast. Voy a cambiar a español, entonces les agradezco muchísimo. Director, thank you very much for your time. We appreciate it from the USFQ and the Institute of Biomedicine, and also Juan Carlos. Thank you very much for all the efforts that you are doing to move this field forward. Aquí en el Ecuador es súper clave el hecho de poder seguir soñando cada vez más alto y mover justamente esas interacciones tan importantes. Quisiera también agradecer a todas las personas que están backstage, a Andrés y a Pierre que nos han acompañado, y a toda la gente de marketing que nos ha ayudado a poder sacar este evento adelante, junto con todos los miembros del Instituto de Investigaciones en Biomedicina que están pues aquí presentes, y a todo nuestro gran cuerpo de amigos y colegas en la Universidad de San Francisco. Entonces, les agradecemos mucho, igualmente a todas las personas que han estado presentes en el broadcast. Muchísimas gracias, Víctor. Muchísimas gracias, Juanca. Muchas gracias a todos.
Gracias a todos. Hasta la próxima.
Gracias. Bye.
Chao.
Bye, Víctor.
Chao.
Bye, Víctor.
Chao, Andrés.
Dreams, they take us places we've never been, show us things we've never seen. Dreams let us reimagine the world we live in. There's a power inside us that can make dreams come true. The power of the genome. We found a way to unlock that power, so you can answer life's biggest questions. Can you find a cure for every kind of cancer? Can disease stop being an inheritance? How can everyone live not just longer, but healthier? These answers are written just 4 letters. We live in a time that is brighter than ever before. And this is just the beginning. This is the genome era.
Illumina first brought NGS to the benchtop more than a decade ago and continues to set the standard for simplicity and speed. The MiSeq i100 series. The series offers two systems. The MiSeq i100 system enables 15 gigabases of data output with 25 million reads. And the MiSeq i100 Plus system has an expanded output of 30 gigabases with 100 million reads. There are 10 reagent configurations for flexible outputs, providing deeper sequencing or the ability to run additional samples for a variety of applications, including small whole genome sequencing, pathogen detection, and targeted gene sequencing. The MiSeq i100 series is simple at every step, from setup to data analysis. The reformulated reagents are shipped and stored at room temperature, eliminating the need for freezers. There's no need to wait to thaw your consumables before starting a run. Run setup is performed in 3 steps. Dilute libraries, add denaturation buffer, load the cartridge. The instrument will walk you through loading the cartridges, minimizing the chance of user error. After a few minutes, the pre-run checks will complete and automatically initiate the run. You can monitor the progress of the run on the instrument or the cloud via a laptop. XLEAP SBS chemistry, our fastest, highest quality, and most robust sequencing by synthesis chemistry, enables a 4-fold reduction in turnaround time compared to the MiSeq system, providing same-day results. Onboard Dragon offers accurate, comprehensive, and efficient data analysis. Data analysis has been simplified with sample-to-analysis workflow solutions, generating results in 2 hours. Through Base Sequence Hub, you will have access to the full suite of Dragon analysis pipelines for accurate, rapid data interpretation and visualization, minimizing the need for NGS bioinformatic expertise. Following the completion of the run, remove the cartridges from the instrument. The cartridges can be easily disassembled, and a majority of the components can be recycled. This is the highest standard in benchtop sequencing. This is sequencing for every lab. This is the MiSeq i100 series.